Dear students, the theme of our lecture, Metabolism of amino acids, digestion of proteins, biosynthesis of urea. The digestion of proteins begins in stomach. Stimulated by food, acetylcholine, histamine and gastrin released onto the cells of the stomach. The combination of these substances cause the release of the gastric juice. Very important components of gastric juice are mucin, hydrochloric acid, pepsinogen. Hydrochloric acid important to create optimal pH for pepsin action and activation. It denaturates proteins, kills bacteria and other foreign cells. Pepsinogen is converted to the active pepsin with the help of hydrochloric acid and enzyme pepsin already present in stomach. Pepsinogen cleaved off to become the enzyme pepsin and a peptide fragment to be degraded. Pepsin partially digests amino acids by cleaving of peptide bond formed by aromatic amino acids. After the stomach, digestion of proteins takes place in small intestine. Stimulated by food, secretin and cholecystokinin regulate the secretion of bicarbonate, enzymogens, trypsinogen, chymotrypsinogen, proelastase and procarboxypeptidase. These proenzymes synthesized by pancreas and secreted into duodenum. Bicarbonate changes the pH to 7. The intestinal cells secrete an enzyme called enteropeptidase that act on trypsinogen, cleaving it into trypsin. Trypsin converts zymogens into active enzymes. It converts chymotrypsinogen into chymotrypsin, procarboxypeptidase into carboxypeptidase, proelastase into elastase, and trypsinogen into more trypsin. Trypsin cleaves peptide bonds between basic amino acids. Chymotrypsin cleaves the bonds between aromatic amino acids. Carboxypeptidase is the exopeptidase that cleaves one amino acid at a time from the carboxyl side. Aminopeptidase secreted by small intestine and cleaves off the N-terminal amino acid at a time. As a result of this enzyme's action, most proteins are completely digested to free amino acids. Amino acids and sometimes short oligopeptides are absorbed by secondary active transport. Amino acids are transported via the blood to the cells of the body. There are different sources of amino acids in an organism. First one, absorption in the intestine. Also proteins decomposition, synthesis from the carbohydrates and lipids. Amino acids used for synthesis of proteins, for synthesis of other nitrogen-containing compounds, as the source of energy, and for gluconeogenesis. Protein turnover, the degradation and resynthesis of proteins. Half-life of proteins from several minutes to many years. Structure proteins usually stable, regulatory proteins short-lived. How can a cell distinguish proteins for their degradation? The special tag that marks proteins for destruction is ubiquitin. It's like black spot, the signal for death of proteins. It's a small protein present in all eukaryotic cells. As for structure, extended carboxyl terminus, glycine, that is linked to other proteins, and lysine residues for linking additional ubiquitin molecules. Ubiquitin covalently binds to the epsilon amino group of lysine residue on a protein destined to be degraded. Isopeptide bond is formed as a result. A special large protease complex, proteasome, digests ubiquitinated protein. This is multi-subunit protease. It consists of two components, 20S catalytic subunit and 19S regulatory subunit. Pool of amino acids that formed in organism used for protein synthesis, for synthesis of different nitrogen-containing compounds, as a source of energy, and for synthesis of glucose, for the gluconeogenesis. There are general ways for transformation and degradation of amino acids. These ways are deamination, transamination, decarboxylation of amino acids. The major site of amino acids degradation is liver. Deamination of amino acids is elimination of amino group from amino acid with ammonia formation. There are such types of deamination as oxidative, reduction, hydrolytic, and intramolecular. The most important for higher animals and for humans is oxidative deamination of amino acids. During reductional deamination, amino acids converted into fatty acids. At hydrolytic deamination, amino acid converted into hydroxy acid. Intramolecular deamination leads to transformation of amino acid into unsaturated fatty acid. L-glutamate dehydrogenase plays a central role in amino acid deamination. In most organisms, glutamate is the only amino acid that has active dehydrogenase. This enzyme active and present in cytoplasm and mitochondria of hepatocytes. Oxidative deamination consists of two steps. In the first step, glutamate, during reaction of oxidation, converted into shift-based intermediate. This is NAD or NADP dependent reaction. In the second stage, shift-based intermediate converted into alpha-ketoglutarate, ammonia splitted from the substrate. The next way of amino acids transformation is transamination reactions. Transamination is a transfer of amino group from an alpha amino acid to alpha keto acid. Usually, alpha keto acid is alpha ketoglutarate. Enzymes aminotransferases or transaminases. Reaction reversible. As a result of this chemical reaction, form new amino acid and new keto acid. There are different transaminases. The most common are alanine aminotransferase and aspartate aminotransferase. Alanine aminotransferase catalyzes reaction of alanine transamination. Alanine reacts with alpha-ketoglutarate. As a result of chemical reaction, pyruvate and glutamate are formed. Aspartate aminotransferase catalyzes reaction of aspartate transamination. Aspartate reacts with alpha-ketoglutarate. As a result, oxaloacetate and glutamate are formed. Aminotransferases funnel alpha amino groups from variety of amino acids to alpha-ketoglutarate with glutamate formation. Glutamate can be deaminated with releasing of ammonia. All aminotransferases for reactions of transamination require the prosthetic group pyridoxal phosphate, PLP, which is derived from vitamin B6. Ping-pong kinetic mechanism consists of several steps. In the first step, the amino group from amino acids transferred to pyridoxal phosphate, forming pyridoxamine phosphate and releasing keto acid. In the second step, alpha-ketoglutarate reacts with pyridoxamine phosphate, forming glutamate. As a result of aspartate transamination, aspartate converted into oxaloacetate, alpha-ketoglutarate to glutamate, and PLP reversibly changed to PMP and to PLP. The next general pathway of amino acids transformation is their decarboxylation. Decarboxylation is removal of carbon dioxide from amino acid with formation of amines. Usually, amines have high physiological activity. They are hormones, neurotransmitters, enzyme of decarboxylation reaction, decarboxylase, coenzyme pyridoxal phosphate. At decarboxylation of glutamate formed gamma-aminobutyric acid, GABA. GABA is mediator of nervous system. Other example of decarboxylation is decarboxylation of histidine that converted into histamine. Histamine is a mediator of inflammation and allergic reactions. During decay of proteins, enzymes of microorganisms in colon or in dead organisms decarboxylate amino acids with the formation of diamines. Ornithine in this chemical reaction converted into putrescine, lysine into cadaverine. After the removing of functional groups of amino acids, the carbon skeleton of amino acids are transformed into metabolic intermediates that can be converted into glucose, fatty acids, ketone bodies, or oxidized by the citric acid cycle. The carbon skeleton of 20 main amino acids are transformed into 7 molecules. They are pyruvate, acetyl-coenzyme A, acetoacetyl-coenzyme A, alpha-ketoglutarate, succinyl-CoA, fumarate, and oxaloacetate. Glucogenic amino acids are those amino acids that degrade to pyruvate or citric acid cycle intermediates and supply gluconeogenesis. Ketogenic amino acids degrade to acetyl-CoA or acetoacetyl-CoA. They can contribute to the synthesis of fatty acids or ketone bodies. Some amino acids are both glucogenic and ketogenic. Leucine and lysine are ketogenic amino acids that converted into acetyl-CoA and acetoacetyl-CoA. Other amino acids marked by yellow color are ketogenic and glucogenic. And last one, marked by pink color, are glucogenic amino acids. They supply gluconeogenesis. Plants and microorganisms can make all 20 amino acids. Humans can make only 11 of the 20 amino acids, non-essential amino acids. Non-essential amino acids for mammals are usually derived from intermediates of glycolysis or citric acid cycle. The other are classified as essential amino acids and must be obtained from the diet. A deficiency of even one amino acid results in a negative nitrogen balance. In the negative nitrogen balance, more proteins degraded than synthesized. At positive nitrogen balance, opposite, more proteins synthesized than degraded. Neutral, it's zero nitrogen balance. Equal amount of proteins degraded and synthesized. During different biochemical processes, an organism formed ammonia. First of all, it released at oxidative deamination of glutamate. Next, during deamination of physiologically active amines and nitrogen bases. It absorbed from intestine as a result of degradation of proteins by intestinal microorganisms. Also, during hydrolytic deamination of AMP in the brain, enzyme that catalyzes this reaction, adenosine deaminase. Ammonia is a toxic substance to plants and animals. It's toxic especially for brain. Normal concentration of ammonia in blood from 25 to 40 micromole per liter. Ammonia must be removed from the organism in form of water-soluble, non-toxic substance. For humans, this substance is urea. It's synthesized in liver and excreted by kidneys. Birds and reptiles synthesize uric acid. detoxified in liver, so necessary specific mechanisms for transport of nitrogen from peripheral tissues to liver. First of all, it's alanine cycle. Glutamate is formed in peripheral tissues in reactions of transamination, but glutamate dehydrogenase is not active in peripheral tissues. Nitrogen transported to pyruvate to form alanine that release into the blood. The liver takes up the alanine and converts it back to pyruvate by reaction of transamination. The glutamate formed in liver is deaminated and ammonia is utilized in the urea cycle. Nitrogen also can be transported as glutamine. Glutamine synthetase catalyzes the synthesis of glutamine from glutamate and ammonia in an ATP-dependent reaction. The urea cycle is the cyclic pathway of urea synthesis, first postulated by Krebs. The urea cycle located in hepatocytes, in matrix and cytoplasm. Urea contains two nitrogen atoms. They come from aspartate and ammonia. Carbon atom comes from CO2. First of all, during urea synthesis, the free ammonia coupling with carbon dioxide to form carboxyl phosphate. Two molecules of ATP are required. Reaction takes place in the matrix of liver mitochondria. Enzyme carboxyl phosphate synthase. Carboxyl phosphate donates carboxyl group to ornithine, the product citrulline. Enzyme ornithine carboxyl transferase. Reaction takes place also in the mitochondrial matrix. Citrulline, after this chemical reaction, leaves the matrix and passes to the cytoplasm. In the next chemical reaction that located in cytoplasm, citrulline, in the presence of ATP, reacts with aspartate to form argininosuccinate. Enzyme argininosuccinate synthase. Argininosuccinate is cleaved to free arginine and fumarate. Enzyme argininosuccinate lyase. Fumarate enters the tricarboxylic acid cycle. During last reaction of urea synthesis, arginine hydrolyzed to generate urea and ornithine. Enzyme arginase. This enzyme present only in liver. Ornithine transported back to the mitochondrion to begin another cycle. Urea excreted via kidneys. About 40 grams of urea excreted daily. Fumarate link citric acid cycle and urea cycle. Fumarate formed in urea cycle enters citric acid cycle and converted to oxaloacetate. What is the fate of oxaloacetate? Oxaloacetate may be used at transamination to aspartate, in conversion into glucose, at condensation with acetyl-CoA to form citrate, and it also used in conversion into pyruvic acid. Disorders of metabolism of amino acids lead to development of different diseases. For example, alkaptonuria, inherited disorder of tyrosine metabolism caused by the absence of homogentic acid oxidase. In this case, homogentic acid is accumulated and excreted in the urine. Urine turns into a black color upon exposure to air. The symptoms that are specific for this disease are darkening of the cartilage of ear, dark spots on the sclera and cornea, development of arthritis. Next disease is maple syrup urine disease, the disorder of the oxidative decarboxylation of alpha keto acids derived from branchy chain amino acids. This disease caused by the missing or defective branched chain dehydrogenase. In this case, the level of branched chain amino acids or corresponding alpha keto acids elevated in blood and urine. The urine has specific odor of maple syrup. Early symptoms of maple syrup urine disease: lethargy, ketoacidosis, mental physical retardation. Unrecognized disease leads to coma and to death. Phenylketonuria is disease that caused by the absence or deficiency of phenylalanine hydroxylase or its cofactor tetrahydrobiopterin. Phenylalanine accumulated in all body fluids and converted to phenylpyruvate. Symptoms are defects of formation of myelin coats of nerves, brain weight below normal, mental physical retardations. Diagnostic criteria are determination of phenylalanine level in blood, test of urine with iron chloride. At positive reaction, formed blue-green color. Also, prenatal diagnostics with the help of DNA.